This is Patrick O'Shaughnessy. I'm a certified industrial hygienist and a professor at the University of Iowa, and I will be discussing data organization techniques in this module devoted to data capturing and organization associated with this InTERACCT series. One goal of this module is to help you understand different organization methods for both time-integrated samples and real-time samples. And the second goal is to provide meaningful interpretation of the results and to help you minimize reporting errors. So in this first of 4 parts of this module, I will provide examples of using a spreadsheet to best organize data obtained for gravimetric analysis of aerosol concentrations. So let's start with the basics. A time-integrated concentration refers to a standard filter-based assessment of a workplace airborne concentration. Therefore, we're going to need mass from a balance and knowledge of the flow rate and sample time to compute the concentration in units of milligrams of dust in the air per meter cubed of air sampled, as shown in the equation at the bottom. So with all that, it seems relatively simple, but how do we go about making that calculation in a systematic fashion, knowing we will need information such as the sample start and finish times and the before and after filter weights? Here is an example of a spreadsheet developed to compute mass concentrations consisting of information, as you can see, in columns A through M and 7 rows. So the spreadsheet is split up into various factors needed to compute concentration. The sample identification, then cells are available to enter sample start and stop times, filter pre and post weights, the flow rate, and then ultimately a concentration based on the equation shown below, which incorporates the net weight of a blank filter. So focusing in on the initial part, it's helpful to incorporate as much information as possible in the spreadsheet. So you need to know the sample location, of course, as shown here in the top left cell A1. And then the sample dates and the sample type are also important. So in this case, a cyclone was used as indicated, and then a filter ID is added. And you can use any kind of numeric and alphabetic notation you want, but in this example, it's S1-7-1 to indicate the first sample, S1, taken on July 1st. And then below you'll see that it's also showing that a blank is part of this analysis. So then moving on to sample time. In Excel, time can be entered as so many hours and minutes. The display of time can be formatted by right-clicking on the cell and then choosing Format Cells and choosing the kind of time display that you want. In this case, it's the simple 24-hour indication of time. As you can see, once start and stop times are entered, the difference between them can be easily computed just by subtracting the stop and start time to obtain the overall sample time. And that will be given in the same time format. To convert that to a number of minutes, the value is multiplied by 1,440 to get the total number of minutes, and as you can see here is 481. Now on to the filter weights portion of the spreadsheet. The assumption is that as an industrial hygienist, you purchase pre-weighed filters from a lab. You then go out into the field and use them, and then you send them back to be post-weighed. So the report you get back only includes the net weight as shown here. If you're going to do your own weighing, then there would be additional columns for the initial weight and the final weight. And as given, both the cyclone weight of 0.254 milligrams and a blank weight of 0.009 milligrams are shown. And given 3 digits after the decimal place implies that a balance with that amount of resolution was used. The final digit is 1/1000th of a milligram, or 1 microgram. And so given this resolution, all 3 digits and only 3 digits should be shown here. So the cells containing weights need to be formatted to show 3 by choosing the format option number and setting the decimal places shown to 3. Also, as shown here, the common practices of compensating for a blank weight is added to the spreadsheet to arrive at an adjusted weight of 0.245 milligrams. Likewise, the sample pump flow calibration should be applied in the typical scenario where at least 3 independent samples of flow rate are taken before sampling, and then another 3 after. From then, an overall average of those 6 values is computed using the averaging function to obtain, as shown here, 2.11 liters per minute. There's always the concern as to whether or not the flow rate has deviated significantly across the sample time. So to test that, a percent difference of pre and post flow rates can be computed and compared to a value of 5%, above which you should probably redo the analysis. In this case, the percent difference is 1.3%, so that is acceptable. With all known information added to the spreadsheet, the concentration can now be calculated. And as shown in the equation, the corrected filter weight of 0.245 milligrams is divided by a sample time of 481 minutes and an average flow rate of 2.11 liters per minute. And applying the correction factors from liters to cubic meters of 1,000, we end up with 0.245 micrograms per meter cubed. Now, just in terms of spreadsheet organization in general, those of you who use Excel will know that you can not only have a single sheet when you open up the file that you have created, but within that file you can have multiple sheets. So something to think about when you have, for example, multiple days of sampling. Do you put them all in one sheet in a file, or do you have multiple sheets in the same file? This decision will also dictate the naming of the file. In the first case, you have one file per sample day. Then your file name should reflect that and have some kind of indication of sample day associated with the file. Otherwise, with multiple samples per file, the file name might be according to the project name. Then you know that within it are all samples across multiple days. Either way, make the file name as obvious as possible so that, say, even 3 months from now, you can look at your file names and understand what it is that is most likely contained within it, to save you grief later on trying to find data. You may have it associated by contaminant type or by different locations within a plant, whatever works for you, but make it as obvious as possible to distinguish one file from another. And this brings up the issue of spreadsheet templates. There's certainly no need to reinvent the wheel, so to speak. So once you have developed a nice template for one sample, you don't want to have to keep recreating that from scratch every time. Once a good structure for your data is made up in a spreadsheet, it can be saved using the word template in the file name and then pulled up as needed. Then you can rename it for the conditions that you are going to apply to it and apply the new data values to it and resave it. In the same way, within a spreadsheet, a completed sheet can be copied as well, and then it is overwritten with new data. And so you can keep tacking on sheet after sheet after sheet within the same spreadsheet, each one developed from the original one in the spreadsheet. Here's an example. So to copy an entire sheet, you can see here that I've right-clicked on a sheet tab called Filter Weights, and there is the option to move or copy. When you click on that, a box comes up allowing you to, quote, create a copy to the book with the file name of grav concentrations in this case. You can tell it to move it to the end of all sheets and click OK. When that happens, you now have an identical sheet that is automatically called Filter Weights 2. And then you can rename that if you want. This is the best way to ensure that everything is exactly transposed into the new sheet within the spreadsheet, especially if there are graphs in the copied sheet. The new graph in the new sheet will refer to data in the new sheet. As you may have found out when copying a graph from one sheet to another, just the graph still refers to the data in the sheet it was copied from. However, the method here of copying the entire sheet, which contains a graph, will ensure that the graph in the second sheet is independent of anything on the first sheet and only associated with those in the new sheet that you've just created. So now we're getting a little more sophisticated, knowing what we would like to collect in a template that may be printed out and is very useful and valuable to the people that are going to view it. Here is an example of a filled-in template for a sheet to compute gravimetric concentration that is put together a little nicer than the previous one shown, which was at a very basic level. In this example, I know that I'm going to go out and sample a site using 2 cyclones and also take along a field blank. So it's set up for that. Otherwise, it's similar in structure to the one already discussed to ultimately compute respirable concentrations. But note in the spreadsheet that entered data is shown in black font and any calculated values are shown in blue font. This visually helps to ensure that a function is not overwritten with an entered value. The template also might be created for a different situation in which perhaps the same worker is sampled multiple times during the day, as shown here. So in this case, we have the same worker sampled twice during the same day. And then we have to compute what's known as the time-weighted average for that person. As you can see, the equation up on the top. So there needs to be an additional column to compute the time-weighted average, which is shown in column P, where the sums of concentration times the sample time for each is divided by the sum of time.
This lesson has been created by the Interdisciplinary Training, Education, and Research Activities for Assessing and Controlling Contaminants from Emerging Technologies Program, a collaboration of the University of Minnesota School of Public Health, the University of Iowa College of Public Health, and the University of California, Los Angeles Fielding School of Public Health. Funding for InTERACCT is provided by the National Institute of Environmental Health Sciences of the National Institutes of Health. The content of this lesson is the sole responsibility of the developers and does not necessarily represent the official views of the National Institutes of Health.